After years of conflicting reports that signals from mobile phone towers can raise various health concerns, a new study claims to provide the first evidence that links the signals with nerve pain. Scientists in the US report that the radio frequency, RF, and electromagnetic signals can cause what's known as a pain response in amputees. They claim they now have scientific evidence to support the anecdotal reports made by people with amputated limbs. The research, published in the journal PLOS One, found that in rats with an amputation-like injury the animals showed clear evidence of pain in the presence of the signals. Dr. Mario Romero Ortega, senior author of the study and an associate professor at the University of Texas at Dallas, said, "Our study provides evidence, for the first time, that subjects exposed to cell phone towers at low, regular levels can actually perceive pain. Our study also points to a specific nerve pathway that may contribute to our main finding. The study is one of the first to look at electromagnetic fields, EMF, in an animal with a nerve injury, in which they report a persistent and significant pain response when exposed to radio frequency and EMF. Rats received either a nerve injury to simulate the pain of an amputation, or a sham operation in which the nerve was not damaged. Researchers believed the damaged nerves would form clumps called neuromas, in which the endings become inflamed and sensitive, and would potentially be more sensitive to stimulation, such as from RF and EMF signals. The rats were then exposed to EMF signals equivalent to standing near a mobile phone tower almost 131 feet, 40 meters, away. Animals received exposure for 10 minutes, once a week for 8 weeks. They found that after 4 weeks, 88% of rats with the nerve injury showed a definite pain response to the signal. Many believe that a neuroma has to be present in order to evoke pain. Our model found that electromagnetic fields evoked pain that is perceived before neuroma formation. Subjects felt pain almost immediately, explained Dr. Romero Ortega. My hope is that this study will highlight the importance of developing clinical options to prevent neuromas, instead of the current partially effective surgery alternatives for neuroma resection to treat pain. Analysis of the animal cells showed that one protein in particular, TRPV4, may play a key role in the nerve-damaged rat's pain response and, said Romero Ortega, is worthy of further exploration. The scientist said sensitivity in humans is often labeled as psychosomatic, and so the phenomenon is not given the attention it requires. Dr. Romero Ortega concluded, in our study, the subjects with nerve injury were not capable of complex psychosomatic behavior. Their pain was a direct response to man-made radio frequency electromagnetic energy.